Lecture 9, the second part of our effort to wrangle the Terra Oceans data into our RStudio session. Today we're going to pick up where we left off last class with the Sunagawa et al. Dataset. Okay, and to make sure we know where we are, here our bioinformatics object is the Terra Oceans dataset, of course. And last class was really about the import procedure. It was about getting that data off of their website and into our RStudio session, preferably using this wget approach, where we don't need to go through our laptop as an intermediary. We go straight to the RStudio session that presumably has a larger disk space, etc., to store large files. And today, this lecture is the key word really is, whoops, I just activated the pizza. Oops, it's really sensitive today. So this lecture is about, oh my, having difficulty writing, sorry about that. This lecture is about tidying data. Now that's really the key word, and it is really, tidy is of course it's the tidyverse, so it's really a key step. Okay, so we'll explain about what tidy means in a bit. But before returning to our deserted South Pacific island as Survivor Man, I wanted to talk about some other mandates for bioinformatics. So a couple of lectures ago, we talked about the role that bioinformatics plays in storing data at one end of the spectrum as a repository, and at the other end in terms of expert databases, curated databases that synthesize that raw data into knowledge. But there are other mandates. One of them is data representation. How we they set standards for how we should describe our experiments and the data that comes out of our experiments. Standards for software and data structures. Well, you know a data structure already. Actually, you know several of them. One of them is the tibble. Now, the tibble didn't come out of bioinformatics. It came out of pure data science. So data science applied to politics or sports or bioinformatics or whatever. And the tibble is kind of a universal data structure. And that term data structure means an informatic structure. It usually has a class for storing data. Okay. But bioinformatics also touches on things like ethics and sharing. So they set policy, the community set policy in these directions. And there's standards for reproducibility. And it's particularly this last one, we're going to come back to it near the end of the course in the last couple lectures to talk about the issues of the reproducibility crisis in science. So we'll talk a little bit more about it today and then more thoroughly at the end. But standards for data representation to start with. Okay, so usually it's about setting a format that everybody agrees upon, that when you upload your data to a database, like let's say the NCBI, then we all agree, they tell us what we need to give. And one of the earliest standards was the so-called Miami standard that you can check out. It's in the, I think it's actually in the recommended reading, perhaps. I'm not sure if it's in the, what do you call it, the necessary reading, although it's very short either way. Let me show, I can just click on it for a second. Yeah, so this is the Miami standard. It stands for minimum information about a microarray experiment. Now, we won't go into all the details here, but suffice it to say is that back in the day of microarrays, the community got together and said, okay, well, if you want to make your microarray data available to the community, then we have to agree about what information you provide. So what kind of microarray was it? What are the samples? What were the preparation procedures for those samples? And also downstream things like what the gene identifiers you used, what version of the human genome database you used if they were human microarrays, and all sorts of other essential laboratory protocol information that other people who wanted to use your data would probably need to know. And so when we submitted our microarray data, we did it in this basic procedure. So now, of course, microarrays are out of date, but there's there's all sorts of um, uh, additional data standards now for other data types, and you can read a little bit about it at FGED. So now for RNA sequencing data sets, you know, there's the equivalent kind of standards. And for example, when you upload to the NCBI, you have to provide a certain amount of information. And you know, this is from one of my papers with my group, and this was a submission to a database called the Gene Expression Omnibus. So we saw, you know, all sorts of sequence databases, whether it's nucleotides or mRNAs or proteins, etc., amino acid sequences. But there's also databases for making your gene expression studies available, right? And in this case here, this was a data set from McGill of about 400 samples for invasive breast carcinoma. And so, you know, they ask you for a summary of what the project was about. They ask you, you know, well, I guess it was 300 samples. And then a lot of extra information I haven't shown you here, who is involved, etc., contact authors, etc. And it's about 2 pages long of information that we have to supply. An interesting idea is the Bermuda policy, and that is part of the required reading, I believe. It's just a short paragraph at Wikipedia. There's another site in the recommended reading. There's a paper about the Bermuda policy that's a bit more extensive, and if you have time to read it, I think it's a good idea. I think we can go to it briefly. So, yeah, it's not formatted too well on the screen here, but hold on a second. Let's see if I can fix that. Where did it go now? Sorry. Here it is. Yeah. So, basically, I think this was in 1996 where a bunch of researchers got together in Bermuda, presumably, and this was at the, you know, well, I would say midpoint of the Human Genome Project, and they were getting prepared for the rules for how the data should be released. So there's a whole bunch of labs participating in the Human Genome Project, and in 1996, the government wasn't clear whether they were going to allow people to patent genes, and I think that's kind of a strange idea unto itself now that we look back. But you know, people were starting to think like they're not going to actually make the sequencing data available because there might be a gene on there that they could patent. And of course, that's almost, it seems to me, a bit, well, it seems immoral, right? That somehow we wouldn't have access to our own human genome and knowledge about our own human genome because a company might benefit from, I don't know, you know, a specific gene on there that it could target with cancer or something. But it didn't work out that way. It took actually until 2001 for Bill Clinton to come forward and say that the U.S. wasn't going to allow the patenting of genes. But the Bermuda Principles were really something very forward-looking at the time, and they were trying to lay down, you know, basically what people who are in part of this project should be doing. And you know, it's for that at the time it was kind of shocking that they should release their sequencing data, anything more than a KB of nucleotides, within 24 hours. The immediate publication of finished annotated sequences. Now that's a really interesting comment, and I think that maybe it was Michelle in one of the discussions while we were talking as a group about why repositories are important, and somebody brought up, I think it was Michelle, that you know, by releasing early we make that data available to the community, and if we hold on to it, then the community doesn't have it. It's just the people who generated it. So the interesting thing here is that we're not trying, the principle was saying, look, you know, we don't really want you to analyze the sequence data. We just want you to release it. In a sense, like, you know, the community will analyze it, right? And you can participate with the rest of the community in analyzing it, but we don't want you holding it on to it for 2 years while you look at it and make up your mind what it means. And the third thing is aim to make the entire sequence freely available in the public domain for R&D to maximize benefits. And I think that's a really healthy approach, right? We shouldn't be making money off the human genome itself. You know, there's lots of things in the human genome that we can, figure out drugs with for specific diseases, etc., or all sorts of other benefits to human health in general, and that's where companies can thrive. But the genome itself really should be something that's public, especially since the government, that is your taxpayer dollars, are what's funding its these sequencing efforts. Okay. So another, for example, is the International Society for Computational Biology. I won't go to it right now, but this is basically something that computational biologists, data scientists, and bioinformaticians, they all belong to. It's a professional society, so we pay a small amount per year to be a member, and they're interested in things about the ethics of computational biology, how bioinformatics and computational biology, data science should be taught, promoting, let's say, gender equality or diversity in these fields, generally promoting the education in universities of these fields, etc., furthering people's interest in the discipline. So they also, they also are basically, they are involved in policy making. And another mandate for bioinformatics relates to reproducibility, and this is a hot topic, and we're going to come back to this, as I said earlier, more in the course, especially at the end. But it's good to have these concepts as we move along a little bit. And there's one society that's now called MAQC, or Massive Analysis and Quality Control. It is actually a computational biology society, and what they're interested in is the issues around reproducibility in large-scale experiments. And probably by now in this course, you're starting to get some insight into why it might be hard to reproduce and make your data and analysis easily reproducible to the community. I ask you, like, if you took your assignment and Aki was to just run it without looking at it, would it work on his computer, right, when he's grading your code? And there's probably things in there that don't work, like paths to files, and maybe even environmental variables, etc. So there's issues that come up with reproducibility that we'll talk about in more detail. But that's also something that's very important, and our society's kind of bioinformatics plays into that, right? And then a little bit more on the technical side, you have bioinformatics involved in trying to set standards for how data is represented in a computer, in a software package, and also standards for both the software itself. So some of these things could be very simple, and we saw an example of the FASTA format earlier in the course. It was part of the required reading. It's just a very simple standard for representing nucleic acid or amino acid sequence data. There's nothing really to it, but everybody agrees on what a FASTA format is, and so if we're going to transmit sequence data between us, we often do it in a FASTA format. If you're really into molecular evolution and phylogenetic trees, then you might use the NUIC format for representing trees in R, or if you're programming in Python or Julia or some other programming language, you still keep that same format so that we can use it across different platforms. Or PDB, protein database files, so for describing protein structure, whether it's coming from NMR or X-ray crystallography, etc. And basically, any kind of biologic information, from basic sequence data to expression data, they all have worked out some kind of format for representing it. And I think we've talked enough about why the tibble is universally a nice idea, that if we all programmed in tibbles, it would be really easy for people to come along and make nice analysis that we could use right away. And the same thing is true about everybody adopting standards for representing data in software. Now, part of this, that while bioconductor is a bit more general than just data structures, it its mandate really is for the analysis and comprehension of high throughput genomic data. So it's broader. It's not just R based. There is some things about Bioinconductor that are Python based, for example, or even BioPerl, etc. I think it's mostly R based, but it's open source and it's open development. Okay, and well, really, it's like this niche of R that's for bioinformatics. And Bioconductor has its own community, led by some really big, famous bioinformaticians, and they're really interested in sort of finding good ways to represent biological data, right? So when a new data type comes out, like single cell expression data, they get really involved with trying to work out the right R data structures to accept that. And if they do that early enough and they do it well enough, and we all adopt, say, a data structure for single cell expression data because it actually is a good structure, then it simplifies everything downstream. And a lot of the code that people write works on everybody else's projects, right? If there isn't such a standard, then of course we're endlessly wrangling data from one format into another format, and then we spend all of our times wrangling, right, and not doing data science. Yeah. Okay, so now Bioconductor actually provides R packages for download, but you install them a bit differently than normal R packages. So I want to say a couple words about that, partly because I think that if you continue to use R even after this course, you're going to run into the fact that some of the packages you want aren't seemingly easy to install in R. To date, we've been using this library function to load a specific package like the tidyverse. So we use this library, and then we call this package tidyverse, and then suddenly all the functions that are available in the tidyverse, and that includes ggplot, dplyr functions for select and filter and arrange, etc. They're all of a sudden they're available to us. So the model here is that the library loads the package from your disk. In our case, it's in the RStudio Cloud server. And basically, before this course started, I put a lot of the libraries we would need, like the tidyverse, into the base package of our RStudio Cloud session. So all of your projects already have access to the disk and the tidyverse library that's sitting on our RStudio Cloud disk. I hope that's understandable. But it begs the question, where did that package come from? I mean, it didn't evolve separately on the RStudio Cloud drive, so it had to come from somewhere. So one thing you could do is look in your RStudio session, and you'll see down, usually in I think this window by default, you'll see here that there's a packages option in the bottom right quadrant. And if you scroll down through that quadrant, you'll see all the packages that are available from the R mothership, basically. Okay, and just by clicking on those packages there, they'll automatically get downloaded from the R servers to your machine or to your RStudio server, right? So there's two different things there. That repository or that centralized site, that's called CRAN. I'll explain that in a little bit. But CRAN is basically a website that everybody puts their R packages on. And so this basically, when you use this package options in RStudio, it's grabbing those packages from CRAN and downloading them to your disk drive. But they're not yet accessible to you in your RStudio session. So CRAN stands for the Comprehensive R Archive Network. Okay, so it has nothing to do with bioinformatics per se. It's all across R. So pure statisticians, biostatisticians, geologists, physicists, anybody who's using R, their packages would go to CRAN. It's not biology specific. Now we could, if I hadn't, actually I think you need your quotes, and that's a little bit of a mistake. But if I was to do an install packages with quote tidyverse, then this function in R, this install packages, will by default go to CRAN. It'll find that package called tidyverse, and it downloads it to your disk. Okay, and now it's ready to be loaded into your system. And then you would do a library tidyverse. Now you don't need quotes, and that loads it from your disk into the R session, okay? And so you've been doing this part, this line here, and I did this line up here before the course started, okay? But you'll find that there are some packages that you need that aren't already in your session, and you'll have to do this install package yourself. As a side note here, if you make your own package, then you can submit it to CRAN, and the volunteers at CRAN, they'll make that package available, and it'll show up in that package list in your RStudio session, and you'll be able to do an install packages, quote, Mike, for example, right? And then globally, everybody will have access to your package. Now, it's a little bit more complicated than that because they enforce rules and standards. They're not very hard to meet, but they definitely look at your package that you submit, and they come back with a lot of suggestions about how you named your functions, how you named your variables, how you documented. They want that you have these so-called vignettes. You might have come across this word in R now. So when there's like an example, not just one tiny example, but a vignette is usually like a kind of like a markdown file that shows you how to use the package, right? It gives you an example and example application with an example data set. So CRAN, in a word, basically, it says, okay, you know, thank you for your package. You know, it looks great, interesting. You know, it definitely, you know, we could publish this from the CRAN website, but make the following, you know, 100 changes, and you're good to go. But they have to do that to maintain compatibility and you know, to make sure that these things and these packages are working properly. But there are other places to get our repositories. CRAN isn't the only place. It's sort of the mothership in a way. But like I said before, Bioconductor also has its own packages that you can download. And you don't get Bioconductor packages from CRAN, you get them from Bioconductor. And I've quoted this quote out because I don't want it to run, because I've already installed but the Bioconductor. So firstly, you would install the Bioconductor package itself, and then you can go and say, well, you know, if you look under available, you'll see that there's hundreds of different packages for everything that you could imagine, from, you know, chromatin accessibility studies to protein structure studies to microscopy. Whatever you're working on, there's probably at least one, if not a dozen, different packages, and then you can install them basically just like this, like before, and then, you know, you can pull them in as libraries. So it's, once you're downloaded, it's not really any different than a CRAN package. Okay, so yeah, so in summary here, there's really several mandates in addition to data that bioinformatics has. You know, data representation, software and data structures, ethics and data sharing, and reproducibility. So we'll talk about some of these things more and more as the course goes on, but that's a good start with those concepts. So now I think that we are going to yeah, right. So now back to where we were. Our original goal for the class was to complete our Tara Oceans wrangling. So last week we saw, we spent some time understanding at some level the Sunagawa data, okay, and then I showed you some Unix, and then we had this notion of a raw folder where we would download the data from somewhere else, and then the wget to import straight to RStudio Cloud. And then we had this really kind of big challenge of how to read into a tibble a multi-sheet spreadsheet, and we managed to do that with a package called readxl, and that was kind of nice. And then we ended the course really with the realization that the data was uploaded as a list, and that's where we ended, and that's where we're going to pick up. So today we're going to talk a little bit more about the list data structure, and then we're going to talk about factors, the principles of tidy data, and some of the functions that we will use to get our Tara Oceans data in final shape. Okay. So now we fly back to our remote Pacific Island, and we can start there. And I'm going to switch over to RStudio now for the rest of the lecture. Cloud, and it's getting pretty windy out here. Okay, so I'm going to work within an R Markdown file, which you guys are getting slowly accustomed to. I like them a lot. So there's a little bit of header here, and title, and HTML document. That's what it's going to output when I push on this button for knit. I'm not going to do that right now because we're going to go through line by line. But in general, with any markdown file, I like to start my first chunk. It's called a chunk, right? As Aki probably talked about. I can label it setup, and I put include equal to false so that it hides some of the execution here, because it's just basically technical due diligence. I usually put my libraries up front first, and then I set my paths. So this is my path in Unix to raw, and this is my path in Unix to the where I'm going to put my refined data, the data that I've processed. In particular, okay, so actually, let's look at that. If I go into the terminal, I can see that I'm in PWD, if you remember, is path, the working directory. I'm in cloud project. That would be the equivalent of here in the GUI. And if I ask for raw, I can do a CD, change directory into raw, and do ls for list. And I can see that I have 3 files here. Remember the 9 sheets Excel file that we started with last week, the README file that describes where I got the data from, because I'm really paranoid about which version and where I got the data, so that when I come back to this data any number of years from now, I'll know exactly what I did. And I've actually downloaded another file that's a bit bigger. It's several megabytes. Just I did this before the class bearing, so we wouldn't have to wait for it to download. And if I go here, I can see in raw, and it's reflecting it there. It's 12.8 megs. It's not very big, but it takes a few seconds to download. I also like to do this, where I say version is 1.0. And the reason for this is that, okay, firstly, I have this file called import.rmd. That's my R markdown file. That's this file. And it's version 1.0. And that's important because it's the code that I used with the data that I downloaded from Tsugawa et al. And if I change something in this code later on, I should update this version later to reflect that. Because, you know, we need to know the version of Tsugawa's data, and we need to know the version of the code that we use to process Tsugawa's data. There's 2 things there. And so this is going to label basically the code, our version of the code. Now, if Tsugawa's data changes, then that data is going to look possibly different when it's processed into our tibble with this code here. Same thing, if Tsugawa's data stays the same, but if we change the code, the table may be different. And we need to be able to reproduce our science. And so what that really means or boils down to is that, you know, you should be able to write code that somebody else can pick up, and they can know exactly what procedure and what data was used. And okay, let's just quickly go to Tsugawa for a second, back to the website that they have for the data. I mean, not to be too critical here, but this companion website tables W1 through 8, you know, I don't like that very much because they haven't versioned their data. Now, possibly because this paper is from 2015, the data is not being changed anymore. However, you know, sometimes people pick out mistakes in papers, and the authors have to write an erratum to the journal and correct it. And so that data might change, right? This data here might change. And so I would prefer that there was a version in the name of the file to say exactly what version I'm using. That's not there, so I'm assuming that that data never changes. So the only thing that can change in this case is our code. So the versioning helps keep track of this a little bit. Now, again, this is a path in Unix, as we talked about, okay? And I'm going to put this into the raw folder we created. So if I go back to project, I put it here. That's where I'm going to download it. And that's, in fact, what I did with this Excel sheet last week. Okay, so we did that. I don't need to do it again. And then we did a little bit of magic here because we, you know, basically searched online for how to download a multi-sheet Excel sheet. So let's get caught up here. I have to go and run this segment here, so I'm gonna just run it so that it goes into there, and so my console window has an up-to-date version with all the variables defined, etc. And then I'm going to load that data again, and I'm going to run it, and that loaded it. I'm not going to go through it again because we did this before. We can make sure, well, we can explore a bit and see that it has the tables there again, and this is basically where we got to last class. We have these this list, right? First import is a list, and if I ask for that, I can see that first import, the class is a list, and I can ask the length of that first import, the length of the list, and it's 9, which isn't surprising because at Tsunagawa et al., this was worksheets 1 through 8, but there was also a README file, if you recall. So that's the 9th. So the first entry in the list is the README, and then the other guys are the individual worksheets. So we can see that if I do first import at 1, this weird notation, I know you don't understand that, I'm going to explain it in a minute, but you can see that that in fact is a tibble, and it looks like the README file that explains that sample label, sample identifier, this in INSCDC run accession number, and it's explaining what the individual fields of these files are. Okay, so the README is there at the first guy, and if I looked at the last guy, first import, the 9th element of the list, I would see that it looks a bit more interesting. I have latitude and longitude and depth and some identifiers for Tara samples, etc. Looks promising, right? Okay, good. All right, well, you know, we don't know the list data structure right now, so I'm going to just get rid of this for a second to keep it. You know, that was the other thing now, is that we should probably get rid of our dependency on Excel. So now I would suggest writing these files, each sheet to a separate file on your file system. And why? Because then now we never have to actually go back to the raw data. We can just start working with the individual sheets in their raw forms. Like, it's still raw. We haven't really done anything to the data yet, but now we don't need to. If we write them as a CSV, and that stands for comma separated values, there's no Excel anymore, and it's going to make our lives easier. Now there's 3 ways to do that. This would be the beginner way, and I would just basically each element of the list from 1 to 9, I would use the write CSV function. That's the first element, and it's going to be sent to this path. Now you could ask, what's this? File path is a nice function in R that makes Unix paths. So I can show you separately what that looks like. I think I just need to copy and paste up to there, and if I put this into the console and run it, you see what it returns is a nice looking Unix path. Cloud project data sheet 1. Now note, I'm saving it now to data and not raw because we've processed the data, right? It's no longer the actual data we downloaded from Tsunagawa. So this is actually going to now go into this folder here called data. So I could do this, and I can basically just write each element of the list, or I could use a for loop, which you haven't seen yet. That's kind of a classic way. The structure is the same here. This is just explicitly writing 1 through 9, and here I'm using what's called a for loop with a variable called named I, and that I changes from 1 to the length of the list, which is 9, and so I first writes out the first element, then the second element, then the third element, and each time calling it sheet 1, sheet 2, all the way to sheet 9. We're going to look at for loops more as we go along in the course. And then there's the expert real R way, which is used to use a function called lapply. We'll see lapplies later on. They have some advantage in R, but let's not worry about those now. This is a perfectly fine way to do this at the beginning, and if I run this chunk, I can see instantly these sheets are now saved as comma separated value files in my project data folder. Okay, so that's some progress. Not huge progress, but a bit. But let's think now about what this, reflect on what this means. That first import is a list, and you haven't seen lists, so let's go through them a little bit. Okay, so lists are a data structure, and data structures is a term often used in computer science. They store specific data in a specific format or structure, and usually that has a specific class. So you've already seen vectors. That's just a line or a row of numbers. A matrix, which is a two-dimensional matrix. You know what a matrix is from mathematics. You have arrays, which are multi-dimensional matrices. And then you have lists, and you've definitely seen the tibble. That's one example of a data structure. A list is another example. It's very flexible. And in some ways it's too flexible. There's no rules. But they're often used for storing unstructured data. Okay, so we've already seen vectors. So here's an example, fib for Fibonacci, a vector of integers 1, 2, 3, 5, 8, 13. Okay, and the thing with a vector or a matrix, but in this case here a vector, is that all of the values have to have the same class. So all of these guys are numbers, right? That's perfectly fine. I could have a vector like this, she loves me not, and it could be a vector of logicals, but they're all logicals. They can be true or false. Or I could have a vector of characters, like here, 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 and here, right? So it doesn't, a vector can have all sorts of different classes. So the data structure isn't really a class per se, but so it can store all types of classes like numbers, logical, or characters, and they're really important in R vectors, especially if you think of a column in your tibble, that's got a certain class, and that's a vector basically, that one column. The tibble itself generally is a matrix, right? It's two-dimensional, but lists, like the tibble as a whole, is two-dimensional. I'm sorry, each individual column is a vector, right? So in general, lists, well, not to say in general, lists are more general. It's yeah, that's really poetic, Mike. So they're more flexible and more general. Okay, so here's an example of a list. Okay, L is, and the way you create a list is just to use the list function, assign it to L, and inside of my parentheses I can have whatever I want. So here, notice that not all of these entries are the same class. Thomas is a character, Pynchon is a character, but 1937 is a number, True is a logical, and Gravity's Rainbow is a character. Okay, so Thomas Pynchon is a famous writer, American writer. Okay, so now what is a list? What does it represent? How is it represented in the computer? And it's like this. It's, think of it as a chain. Okay, so each element of your list, right, it has this is sort of tethered off of this support, and so this guy goes down here, and this element here is a character, and it's linked to the next guy that's holding or tethering a character, that's linked to this guy that's tethering a number, that's linked to this guy that's tethering a logical, then character, right? Okay, so the class of L would be list if I was to put that into R. The length of L here is 5, 1, 2, 3, 4, 5, and there's no names for this list so far. I'll explain names in a second. But now now this is a tricky thing, okay? This is definitely tricky concepts that you won't get right the first time through. But it's important that you try to understand a little bit. So if I write now square brackets, okay, so in computers we have parentheses like this. That's always for a function. I think it's always a function in R. These brackets are for indexing into a vector or an array. We also have curly brackets, right? And they're used in R. Maybe I have an example of a curly bracket. Yeah, I do. we're going to see lots of curly brackets later. For example, here in our for loop, those curly brackets, or curly braces we'd say, not brackets but curly braces, they delimit a body of code, but that's a little bit for later on. We don't need it now. We're going to get there. Now where did my good notes go? Okay, so back to here. Now if I want a sublist of L, I could ask just for the third element, and that would get me back 1, 2, 3, this guy here. If I ask the class of that guy, it's still a list. It's just a smaller list. I could ask for all of these guys, cut these guys out, and just take the remaining 3, and that would be L, single bracket 3 to 5. Now, if I ask the class of this guy, it's still a list, right? It's just smaller than the original L. I could also, if I wanted 1, 2, this guy and this guy, but not the third, minus 3, I could write, and I made a mistake here. I made a mistake. I'm sorry. This is incorrect. When I forgot to delete this. Whoops, I don't want that big giant guy. That guy would go away, okay? And what I would have is that guy will go to, let me see here, 2, 2, 2. Okay. It's not very nice that I had this thicker line, but you get the idea, okay? We'll leave it at that. So that guy, that minus 3 chops out the third element, but it's still a list, right? Okay. Hopefully you're all following there. Okay. And again, the fact that this line is thicker than the other ones is just because I forgot to change the thickness of my my pen. Okay. All right. So if I go back to L here, and I ask for L1, I'll get back just the first element, right? Like this. Now it's still a list, right? It's still this value, right? This character value is dangling or tethered from the list, this backbone. That's a good name for it, is a backbone, but it's still a list. If I ask for L at double bracket 1, I get back the value at that element of the list. Okay, so L double bracket 1 goes here and then down and returns this. L single bracket just returns this. So this guy's down here and what's tethered from it. Okay, so if I ask the class of this now, of L double bracket 1, it's going to be of class character. If I ask the class of this guy again, it's going to be of class list. And what about if I said something like this, L double bracket 1 to 3? That actually shouldn't work, right? That shouldn't be allowed, because it's asking, you're trying to ask for 3 elements here, but the problem is that this guy is a character, this guy is a character, and this guy is a number. So you can't really return data of 2 different types except in a list. So if you type this into R, what you're going to get back is just a list. If you want, you can't really do that. You would have to do something like, if you wanted the 3 elements, you would have to write out, let's see here, sorry, get my pen. You would have to write L double bracket 1, L double bracket 2, and L double bracket 3. And that makes total sense, because what you're doing is getting this guy, and then the second command is getting this guy, and the third independent command is getting this guy here. Okay, but the nice thing, well, there's lots of nice things about list, because now the power really is that you can dangle anything you want off of off the backbone, right? After, I guess, what's the individual things in a backbone? Your discs, right? They're like spinal, each individual spinal disc. That's not too elegant. Okay, but you know, it can get very confusing to have to remember that L at 3, L at 3 is the year of birth, right? And L at 4 is whether he won a Nobel Prize or not. Actually, he didn't win a Nobel Prize. It's false. He deserves a Nobel Prize, but he's never won a Nobel Prize for literature. But maybe one day. So, okay. So, what you can do is you can name lists in elements of a list. So, I can ask for the names of L, and it'll be null at the beginning. You can check that for yourself. If there's nothing there, if you ask for the names of an empty list, I'll show you right now. Here, let me just create a list really quickly. Let me say L assign list 1, 2 things, A, right? If I ask for the names of L, the names are, it's just null. There's no names. Okay. But then I'd have to remember what each element is. So instead what I can do is execute names at L is set equal to, well, first name, last name, birth year, Nobel literature, and novels, right? So now, once I've done that, what happens is that I can, now, why can't I scroll up? There we go. I can do things like this. L $ birth year, when I access that, will give me back the same thing as L double bracket 3. So L $ birth year will give you back this value here. Not this. That would be L single bracket 3. But if I want the value there, I can use L $ birth year. Likewise, I can say L $ novels, and I'll get back here, which is the same thing as asking for L $, should be, make sure that everybody can see that. This is a 5, right? Okay. All right. Okay. So that's kind of nice. It really helps to kind of organize your list so that you're not using indexes and you're using natural language to reference the information in the list. That's really important when you come back to your code a few years later, because you're never going to remember what element of the list had the year and what element of the list had whether they won a Nobel Prize or not. Okay, but lists can have anything in them. They can even have lists, right? So I could change a list so that one of its or more of its elements contain a list within them, and those lists can contain lists, and you can put anything you want. You can even put functions in a list, all sorts of things. So Pynchon's written many novels, so I could actually modify this so that it's not one novel that's hanging from novels, tethered from there, but all of his novels. So how would I do that? Well, I would just add in here a list, and now this list, this is tethered to a list, and the first element of this list is one of his novels, Crying of Lot 49, and the next one is Gravity's Rainbow, and the next one is Vineland. Okay, so now that's a little bit more useful per se, right? Now I can store more information in a nice way. So now if I want to access all of his novels, I could ask for L $ novels, right? This would be the same thing as asking for L double bracket 5. So this would be the same as L double bracket 5, right? Okay. That's really not very clear, so I'm going to just write that again. So L double bracket 5, okay? And now L $ novel, the class of L $ novels is a list, right? So that list now has 3 elements, and if I just wanted the second and third, so these guys, I could just ask for L $ novels 2 at 3. If I wanted the name itself, the character of a second novel, that's this, right? Not this, but this, I would ask for L $ novel double bracket 2. Okay, so again, this guy here, that's going to correspond to this part here, right? Okay, it's going to access those 2 parts. This guy here this guy is going to access just that down there. Okay? All right. In fact, well, you know, I mean, there's more than one author in the world, so I could make a list of lists of lists. So, for example, here, the first element is what you just saw in this list. This guy here, the first element of is going to be Thomas Pynchon, 1937, true, and then the list of all of his novels down here. Okay, that's for Thomas Pynchon. But the second element of this list is going to be for a second author, let's say Mordecai Richler, 1931, false, and then his famous novels down here. Solomon Gursky Was Here, etc. Barney's Version, and a number of others that he wrote. And if you're new to Montreal, and whether you might be Canadian, but new to Montreal, or whether you're from a different country, I highly recommend reading Mordecai Richler. He lived in basically Mile End, I believe, probably somewhere between the border of Mile End and the Plateau or Outremont. I don't know exactly where, but in that general area. And he was a Jew in Montreal in the I guess probably like growing up in the 50s, and yeah, he writes a very colorful, interesting perspective of what it was like to live in Montreal at those times, and to be Jewish, and they're really funny novels too, right? I mean, I don't think he's ever received really the respect that's his due, and well, we don't really celebrate him here in Quebec, and I think it's perhaps partly because he was an English writer, which is a bit unfortunate because he has very good novels. But anyways, it's a very interesting perspective on Montreal, and I highly recommend it. In fact, well, he's not a writer, he's a computational biologist, but David Sankoff, I'm not sure if any of you have heard of David. He was professor at UDM for a long time, and now he's at University of Ottawa. Okay, he left about maybe, well, I guess close to 20 years ago, 15 years ago, to the University of Ottawa. David, he grew up also in Montreal, at, well, I think, perhaps at the same time as Mordecai Richler, maybe a bit, he's probably a bit younger than what Mordecai was. Well, I'm sure he is, but he'd probably be very offended if you heard me talking right now, but I don't mean any offense. He's a great person to talk to. Okay, firstly, he's maybe arguably one of the first computational biologists in the world, and the reason why Montreal's been so strong in computational biology for so many years. I think he entered computational biology from linguistics, computational linguistics, and I think he was studying Quebecois language, the structure of it from a computational perspective. And he's a great person to talk to. He's been very supportive of young computational biologists in the country, especially women. He's been really promoted them and really got them involved and part of the community, and made sure that they're included. In many cases, we can point to lots of people, but in generally all the young professors, he's always been there to review and to support us and give us advice. He has great stories of what it was like to grow up in Montreal as a Jew, and it used to be in McGill, for example, that they would only allow in like one Jew per year to be a lawyer or a doctor or something like that. I'm not exactly sure. And it stopped amazingly late, like in the 60s and 70s, when these things finally stopped, which seems just crazy. But yeah, a bit of Montreal history. Okay, so, but David Sankoff, he's definitely, yeah, famous guy, right, from computational biologists at least. All right, so back to our tibble, our prototypal. Okay, so now we have a little bit of hope to understand. Now you see what I'm doing here. If I scroll back up, first import double bracket 1 is referring to the first sheet, right? And I'm asking for the information that's in, and it's actually a tibble, right? In that list is holding tibbles or dangling 9 tibbles off of it. All right. So here I was just exploring a little bit what's in this first import. We can run it and just check it out. And well, T1, I just made it for ease of writing. That's table 1, because remember that first import at 1 is the README file. And if we look at T1 here, yeah, that's going to be a tibble. It didn't show up here. So, well, you know, the nice thing is to use the view function, because you get this spreadsheet-like perspective of the columns. Oh, sorry. Oh no. I'm going to pause and reset everything up. Somehow that crashed my session. Okay, we're back. Yeah, so I fixed this crash. So you can see here that the observations are all in the rows, and there's quite a few rows, and my variables are along columns like this, right? So you get kind of an overview of what was in that Excel sheet, our first glimpse of it. Okay, so that's the view function. Now, we talked last class a little bit about heuristics, and one nice thing about Reader, which is a part of the tidyverse, is that it has these nice heuristic parsers. So they try to figure out what kind of data is in your the incoming data. So if I run guess parser on the station identifier of T1, I see that it's a character, and that makes sense, right? So I come over here and I come over to station identifier. Where is it here? There it is. It looks like it's a string, right? A character, right? So it got that right. It has a number in it, but it managed to get that right. And if I go back over to here to import, if I guess on date time, I do this, right? It thinks that it is date time, and that makes sense. So if I come over here to T1 and I find date time, I see that here we are. That looks something like this, and it's figured out that that is the date and time for each observation. And in fact, in R we have a date time class. Okay, so it holds the date and time. Okay. Now we want to get this tibble T1 into a tidy format. So we have to review what it means to be a tidy tibble. Right? It's kind of a funny word. So I'm going to switch over here to explain the principles of tidy now. So there are 3 interrelated rules here, which, if they're true, all simultaneously, we'll call the data set to be tidy. So in here, each variable must have its own column. Okay, so in our case, this is sample label, for example, the accession number, etc. They all have to have their own column, and each observation must have its own row. Okay, so in our case here, what's an observation? And basically, if we go back to the Tara Oceans project, each observation is at some given site. That's really this sample label here. It's all of these things about taking a sample at a specific latitude and longitude at a certain depth, you know, what the environmental features are, and then all sorts of variables that are collected about where exactly and what kind of environment that sample was taken. Okay, that's one observation. So each such observation has to be in a row, and then each value must have its own cell. So we don't want to have a cell, for example, having 2 or 3 values in it simultaneously. Okay, that seems probably very easy. Like you'd be saying, like, you know, but Mike, every data set is all, you know, has to be kind of in that tidy format. And the answer is like, no, wait, you'll see that actually very few data sets are in that satisfy all 3 of those rules. So basically, the process of tidying, which come back to the beginning of the class, Remember I said that tidy was going to be the key word today. The first step is always to figure out what the variables and the observations are. Okay, so that's, you have to consult with who generated the data. For us, we have to think about the what they wanted to do in the Tara Oceans. So I would say here that our observations is measurements at a specific site in the ocean, latitude, longitude, etc., at a certain time at a certain depth, and whatever other parameters might be that they took. So they know that they went in with at a certain time, a certain place, at a certain depth, etc., and they took a sample. Now they might have done different depths at that same latitude and longitude at the same time, or maybe they did another sample a day later. That would be a different observation. Now, so observations are your rows. Now your variables, well, I don't know all of those variables, but there are going to be things like the nitrate concentration, the oxygen concentration, the temperature of the water. All of these things are different variables or attributes you measured about that site. Okay, so that's what our goal is going to be. There's going to be, for each sample that we took, or Tara Oceans took, is going to be one row. And each column is going to be all the different variables that we measured about that one site. Okay. So you need to start thinking about the Tara Oceans data, and we're going to go through that. But let's back up to TCGA for a second, okay? And look at TCGA for 1 minute to go back to our old data set. So remember we had a participant like A1NF, if you recall that, okay, in our tibble. And would this be tidy data if we had this column here called ESR1 expression at time of diagnosis, might be 628, and we have another column that's ESR1 expression after treatment? Is that one, is that tidy? And I would argue that it's not tidy, because ESR1 is a variable that we measure, okay, and at diagnosis or after treatment is a conditional on that, right? So that wouldn't really be considered two separate variables. Now it's the same object that's being measured, but at two different time points in this case. Okay, in the book they have an example of Afghanistan and population measured at two different years, 1999 and 2000, and they say basically the same thing. It's not tidy because 1999 and 2000 are conditionals on the same measurement, which is population. So to make this table tidy, we would use the so-called pivot longer function. So here our tibble t is piped into pivot longer, so that means that t is here at the beginning implicitly with the pipe. And what we want to do is we want to pivot to make longer on at diagnosis ESR1, that's this column, and after treatment ESR1 here. And we're going to put those into a single measurement point, okay, and that's going to be called names2. All right, it's going to have two different values, either at diagnosis or after treatment ESR1, and the values are going to be the expression of ESR1. So if we do that, what we're going to get back is something that looks like this, but now, where's my pen? I'm going to have a participant. Let me make my line a little bit thicker here so that we're sure everybody can see it. Okay, participant, okay, and now I asked for something called measurement point. That's that guy. And a third column that's called expression. That's what my new tibble is going to look like in structure. And now the values are going to be A1 and F. And this is going to be something like at diagnosis ESR1, okay, and this is going to be 628, and this is going to be A1 and F after treatment. Oops. After treatment at 527. Okay, so now that might seem like, well, what do you think, right? I mean, you might want to pause and think about this a little bit, because there are some issues here, right? Because now I've switched this measurement point here to be rows. So now you might come and say, but now, Mike, you know, your patient or your patient sample is observed twice. So we have to be careful here. Each row is not a patient, right? Each observation is not a patient. It's a sample from that patient. Another example that you've already experienced is the a column called tumor, right? Remember the tumor that we looked at when we were, well, you looked at in the midterm too, and the values here were either tumor or normal, right? So we know that, for example, one patient might have at least two samples in the set. One's coming from a normal tissue from that patient, and one from the tumor itself. So we have to be careful here that our observations are not patients. Our observations are, in fact, our observations are samples, right? So we have to be careful a little bit there. All right. I hope that you understand that. But we've been talking in class too. Okay, so the opposite way of that, longer is wider. So it's the other direction. So one observation across many rows. Now, that here, right? We're saying, okay, like, you know, you gotta really stop the video and think about this, right? Because I still, well, I mean, not to be arrogant. I mean, I've had more practice at this, and I still have to stop and really think about this for a while. Maybe take the dog for a walk, etc., and, you know, kind of let it mull over in my head for a bit. So I have here the same participant, two samples, and the gene is ESR1 in this row, and then aniline is in this row. So, you know, for that sample, these are two different variables, right? And I'm putting the variables on a row. So that's, you know, it's a little bit philosophic, but really ESR1 and aniline are two different variables. You might then say, well, let's go back, but aren't these two different variables? And you know, not really. They're both measuring ESR1, but at different time points. Now you might say, well, that's a bit esoteric, right? But it is a little bit maybe debatable, let's say. And I think ultimately, what you know, tidy is what you think, right? You know, it's not a strict definition. It's just that you think about these things of what your variables are and what your observations are. So we probably, though, should change this to what is a more tidy version. And if we did that, we would take our tibble T here, and we would pipe that into pivot wider, so the T is implicitly here. And now names from is gene, okay? And names the values from is from this guy here. And I hope that did I draw this out? Right, I didn't draw it out, so I actually meant to put it right here. So that, what's that table now going to look like? Now, what that should look like is something like this, where I have I've gone from gene to values from count, and so what I should get is participant as before. I'm not going to write it all the way out, and now I'm going to get something like ESR1, and this is going to be aniline, okay, the abbreviations, and now my counts are now I can fill in my counts with individual rows. So now, okay, so you understand that this names from gene here. It took this gene, which has these two different values, and it's made those now variables, okay? And now it's saying, take these values here and put them in. So now if I go back to here, it's going to be, well, A1 and F. as before. ESR1 is going to be 628 here, and that's going to be 527. And now that tibble looks like what we used before when we were actually in our small bracket tibble.